It's 6 o'clock.
Thank you, computer. That was not scripted. So what we have here is a new N-terminus because we've separated the ER localization signal from the rest of the peptide sequence. Remember, the N-terminus is not the creation of any kind of new group or addition of any new group. We just always have one N-terminus and one C-terminus on a polypeptide chain, and so this is the new N-terminus that is created by cutting off the signal sequence, which was at the old N-terminus. So this protein now is going to be folded in the ER. It might associate with chaperone proteins in order to fold correctly. It might get sugars added onto it, and it will be transported to where it's supposed to go. So this protein might have other signal sequences in it. So I've highlighted a couple of areas that might have localization signals for other areas. So for example, if you look in your textbook at that chart that has the amino acid sequences for the localization sequences or signals, the mitochondrial signal is at the N-terminus as well. So how does this happen? Well, first, the proteins that are going to eventually end up in the mitochondria are translated at the ER. And then we cut off this localization signal to make a new N-terminus, and that is going to be where the mitochondrial localization signal is. Maybe this isn't a mitochondrial protein. Maybe we don't have this signal. Maybe we have a nuclear localization signal. If you look in the chart in the book at that, then the nuclear localization signal is going to be in the middle of the protein. It's not at the C or the N-terminus. My point is that you could have additional localization signals in the single protein. Even if it doesn't have a job in the ER, this just means that it needs to be processed in the ER for folding or addition of sugars, or insertion into a membrane. So this is the other possibility. If we go back quickly to here, because I'm running out of battery, so we're translating this protein again, reading the mRNA. If this protein is a transmembrane protein, then it's not going to just be spit into the ER lumen. That is the technical term for it, spitting. Just kidding, it's not. Okay, this is my phone on a Friday night. So this protein is going to end up in the membrane. So what happens is this ER localization signal still gets cut off because we're still breaking that peptide bond. So we are again generating a new N-terminus. This is the old one. We have a new one over here. So you can see that this there's no bond between the rest of the peptide and the original beginning of the peptide. But there is also a signal over here which is called the stop transfer sequence, which is detailed in your textbook. Again, I just drew a simplified version of the illustration in your textbook here. And so this sequence is going to be a hydrophobic sequence. It's going to have those hydrophobic amino acids, non-polar, and it roots the protein in the membrane. So this is hydrophobic. It's going to stay in the membrane because it's nice and comfortable there interacting with the hydrophobic tails. And then the two ends of the protein will be hydrophilic. They're happy interacting with the hydrophilic heads. Now this protein is now a transmembrane protein. After the stop transfer sequence, all that does is to tell the ribosome that after that sequence, perhaps this is the stop transfer sequence here. We're making this messy, but that's okay. So this is the stop transfer sequence, and it tells the translocator protein, don't let any more peptide sequence through here. So it stops, and the ribosome is going to continue translating, but in this case, you're going to have the rest of the peptide chain out here. So that's how you end up with some of the peptide chain out here. Perhaps it's a little bit longer than this. And you're also going to have the original part of the peptide chain, which was translated down here, and then the stop transfer sequence. And then, of course, if this peptide needs to go to another place in the cell or in the cell membrane, then it's going to be transported by a vesicle, so this whole area of the membrane is going to be moved. And that's it. I'm going to make another short video about the diagram I drew on the chalkboard on Wednesday.